Hello, I'm Maruk Said, your microbiology guide. Let's explore in-depth micro techniques and uncover groundbreaking insight together on this educational journey into the fascinating realms of microbes.
Streptococcus pneumoniae, also known as strep pneumonia, can be broken down into its components. Strepto, meaning chain, coccus, referring to a round shape, and pneumonia, indicating its role in causing pneumonia. These are round bacteria that typically grow in chains and are often found in lancet-shaped pairs called diplococci. Streptococcus pneumoniae is the leading cause of community-acquired pneumonia, which is pneumonia contracted outside of a hospital setting. Now, let's dive into the anatomy and physiology of Streptococcus pneumoniae. These bacteria have a thick peptidoglycan cell wall, which absorbs purple dye during gram staining, classifying them as gram-positive bacteria. They are non-motile, do not form spores, and are facultative anaerobes, meaning they can survive in both aerobic and anaerobic environments. Additionally, they are catalase negative, indicating they do not produce the enzyme catalase. When cultivated on a medium called blood agar, Streptococcus pneumonia colonies cause alpha hemolysis, also known as green hemolysis. This is because they produce hydrogen peroxide, which partially oxidizes the initially red hemoglobin in the blood agar to create green methemoglobin. Other Streptococcus species, such as Strep viridans, are also alpha hemolytic. To distinguish Strep pneumoniae, an optochin test is performed. In this test, an optochin-containing paper disc is placed on a plate of blood agar, inoculated with the bacteria. For Strep pneumoniae, the growth of the bacteria is inhibited in the zone surrounding the optochin disc, indicating optochin sensitivity. In contrast, Strep viridans are optochin resistant, meaning there is no inhibition of growth around the optochin disc. Another test that can be used to distinguish Strep pneumoniae from other alpha hemolytic Streptococci is the bile solubility test. This test can be performed in a test tube, or more commonly, by dripping drops of bile onto bacterial colonies. Strep pneumoniae is bile soluble, meaning that bile salts will promote the lysis of the bacteria. In contrast, all other alpha hemolytic Streptococci are bile insoluble or bile resistant. Strep pneumoniae has several virulence factors, akin to assault weaponry, that help it attack and destroy host cells and evade the immune system. Firstly, Strep pneumoniae is encapsulated, meaning it is covered by a polysaccharide layer called a capsule. This capsule features pili and fimbriae, which are hair-like extensions that aid in attaching to host cells. Once attached to a mucosal surface, such as in the nasopharynx or the middle ear, Strep pneumoniae can multiply and produce biofilms. A biofilm is a layer of gooey material composed of exopolysaccharides, EPS, within which Strep pneumonia bacteria live and reproduce. If you compare a biofilm to strawberry jam, the seeds would represent the bacteria, while the rest of the jam would be the EPS. The bacteria within the biofilm hide from the host immune system and antibiotics, and can even exchange resistance genes, making them particularly sneaky. Another virulence factor is pneumococcal surface protein A, PSPA, which inhibits complement activation. This allows Strep pneumoniae to evade opsonization and subsequent destruction by the immune system. As if that weren't enough, Strep pneumoniae also produces toxins, with the most important being IgA protease. IgA protease destroys immunoglobulin A, IgA, which normally binds to invading bacteria, allowing neutrophils to destroy them. This is akin to a computer virus disabling antivirus software. Another virulence factor is autolysin. Activation of this enzyme causes the breakdown of the bacteria and the release of its internal components. These released components further activate the host immune system, thereby enhancing local inflammation and leading to the destruction of host tissues, such as alveolar capillaries and pneumocytes. One of the components released from the bacteria is pneumolysin, which is another virulence factor. Pneumolysin is a toxin that binds to cholesterol in cell membranes and forms pores, ultimately leading to cell lysis. The most remarkable element is that strep pneumoniae may quietly colonize the nasal canals and sinuses without causing harm, as long as the immune system keeps them in check, limiting their development and preventing them from moving to other regions of the body. Individuals with weakened immune systems, such as babies and the elderly, are more vulnerable to illness. HIV infection, diabetes, cancer, and alcoholism are all immune-suppressing diseases. Furthermore, certain hazardous chemicals and cigarette smoke might impair local respiratory defense mechanisms, rendering people more vulnerable to strep pneumoniae infections. Strep pneumoniae can cause a variety of illnesses. One example is rhinosinusitis, which occurs when the mucous membrane lining of the nose and paranasal sinuses becomes inflamed, resulting in symptoms such as fever, face discomfort, and headache. Strep pneumoniae can also enter the eustachian tube and cause middle ear infection, known as otitis media, resulting in discomfort and earache. Chronic otitis media can go to the mastoid antrum behind the ear, causing mastoiditis. Strep pneumoniae can cause meningitis by spreading the infection from the mastoid antrum to the cerebral cavity. If you have watched till here, don't forget to subscribe to Family of Science Lovers. Press the bell icon button so you are always updated. The most prevalent illness caused by strep pneumoniae is pneumonia. Strep pneumoniae clings to alveolar cells, or pneumocytes, and produces pneumolysin, which destroys pneumocytes and alveolar capillaries. Pneumonia symptoms include fever, chills, chest discomfort, and a cough. In extreme situations, strep pneumonia can penetrate the alveolar capillary wall and enter the circulation, causing bacteremia. Bacteremia can eventually develop to significant complications such as sepsis, which can result in organ hypoperfusion. Sepsis induced by strep pneumoniae is more likely in those who have spleen issues, as the spleen is vital for protection, particularly against encapsulated bacteria. As a result, strep pneumonia infections are usually more severe in people who have had a splenectomy, spleen removal, or in sickle cell disease patients who have functional asplenia. If you have watched till here, don't forget to subscribe to Family of Science Lovers. Press the bell icon button so you are always updated. Once in the bloodstream, strep pneumonia can cross the blood-brain barrier, invade the central nervous system, and cause meningitis. People with meningitis develop symptoms such as fever, neck stiffness, and headache. On rare occasions, when the walls of the paranasal sinuses are very thin, strep pneumoniae can enter the cranial cavity and cause meningitis. Strep pneumoniae can also cause pneumococcal endocarditis, forming clumps called vegetations that damage the heart valves. It can infect the pericardium, resulting in purulent pericarditis, where pus collects in the pericardial sac. Additionally, strep pneumoniae can spread to a joint, causing septic arthritis, or cross the blood-brain barrier, and cause meningitis as a complication of bacteremia, rather than as a primary disease. Once it has crossed the blood-brain barrier, it can also lead to an intracranial or spinal epidural abscess. Finally, strep pneumoniae can spread from the bloodstream to the peritoneum, causing spontaneous bacterial peritonitis. This is more common in individuals with cirrhosis who develop ascites or excess fluid in the peritoneal cavity. Diagnosing a strep pneumoniae infection involves culturing biological samples such as pus, sputum, cerebrospinal fluid, or peritoneal fluid. Alternatively, PCR can be performed to detect strep pneumonia DNA. Individuals at risk of strep pneumonia infection should get vaccinated. This includes young children under 2 years old, people over 65 years old, and those with chronic diseases such as diabetes, HIV, and cancer. It is especially important for individuals with sickle cell disease and those with functional asplenia to receive the vaccine. If you have watched till here, don't forget to subscribe to Family of Science Lovers. Press the bell icon button so you are always updated. Currently, vaccines against strep pneumoniae are divided into 2 main groups based on their mechanism of action. The first group is the pneumococcal polysaccharide vaccine, PPV-23, which contains 23 purified capsular antigens and is primarily recommended for adults. The second group is the pneumococcal conjugate vaccine, PCV13, which contains capsular polysaccharides specific to 13 strep pneumonia serotypes bound to a non-toxic recombinant variant of the diphtheria toxin called CRM197, serving as a carrier protein. PCV13 is recommended for young children. Finally, treatment for infections with strep pneumoniae requires antibiotics. The classic first line of treatment includes beta-lactam antibiotics such as penicillin G and V or cephalosporins. However, strep pneumoniae has been progressively developing resistance to penicillin and its derivatives, such as amoxicillin. As a result, alternatives include fluoroquinolones, the combination of amoxicillin and clavulanic acid, as well as third-generation cephalosporins like ceftriaxone and cefotaxime. Ultimately, vancomycin and linezolid can also be used. All right, here's a quick recap. Strep pneumoniae is a gram-positive round bacterium that lives in chains. They are non-motile, non-spore-forming, alpha-hemolytic, optochin-sensitive, and bile-soluble. These bacteria can asymptomatically colonize the nasal cavities and sinuses, but they can take advantage of a weakened immune system, particularly in very young or very old individuals, or those with diabetes, HIV infection, cancer, splenectomy, or sickle cell disease. They cause diseases such as rhinosinusitis, otitis media, pneumonia, and meningitis. The treatment of infections with strep pneumonia involves the use of penicillins. However, in the case of resistant strains, fluoroquinolones, third-generation cephalosporins, and vancomycin can be used. Thank you so much for watching. Don't forget to subscribe to my channel, press the bell icon button. Thank you!